Hello everyone, I'm Xinyu. In this presentation, I'll talk about protein analysis of environmental response of E. coli, the determination of unknown treatments. So from the experiments we've performed, peptide fragments were run in the LC-MS, and the information of them need to be identified. First of all, I'll introduce the tools that helped me getting further information of protein change. PicStudio's software is the first choice to use as it is one of the suggestions that can provide sensitive and accurate analysis we may need, which includes de novo sequencing to identify novel peptides, database search to identify the database peptides, Spider to find the peptide with PTMs, in chorus to increase coverage by using multiple search engines. And the next one is the UniProt website. UniProt is a freely accessible database of protein sequence and functional information, which contains a large amount of information about the biological function of proteins derived from the research literature. And what I have used from this website is the retrieve or ID mapping for the protein names, gene names, and the biological processes. The last one is KIC pathway database, which is the writing diagram database. It's the core of KIC resource. And the aspect that I have focused on is the cellular processes. Here shows the protein profile bitmap generated by PicStudio. And the UniProt is the data resource I have used to infer biology. This heat map indicates protein experiencing changes between controls and unknown treatments by using LC-MS label-free proteomic analysis. The level of the color represents the degree of protein upregulation and downregulation when the unknown treatments and the controls are compared. And the content in this table is the most useful information that I have sorted out for determining the cause of protein change. Among these proteins, the most inviting type is the cold shock protein or cold shock-like protein. Their gene names are CSPA, CSPE, and CSPG. CSPA and CSPG are the primary cold shock proteins of both pathogenic and non-pathogenic E. coli. CSPA is remarkably induced when the temperature shifts from 37 to 15 degrees Celsius, and CSPE constitutively expressed at both higher and low temperature. Therefore, we believe that the occurrence of CSPA and CSPG is more valued than CSPE to determine that the treatments were affected by cold stress. Acid stress chaperone (HDEB) usually appears at pH 4 to 5, under which environment the protein maintains a well-folded dimer. For genes TAR and TSR, we found that the appearance of the typical proteins is attractant and repellent in KIC pathway. As the pathway graph here, serine leads to the appearance of TSR, and aspartate leads to the appearance of TAR. As a conclusion, the unknown treatment is possibly the cold stress, which is under room temperature. Besides, weakly acidic substances may be added to the treatment, that the pH can be around 4 to 5. Attractants such as serine and aspartate amino acids may exist in the treatments. This is the end of my presentation, and thank you for your listening.